Okay, here we go with chapter 26, section 2, which is on sponges. This is a really important section. I'm just kind of zoom in on the course website here. Topic 8, as I have it arranged here now. Sponges are the first example of cell specialization and multicellular life for animals. When we step past the protists and we look at colonial cells which come together, Mother Nature was hinting at multicellular life forms. And just like you and I, the cells in different parts of our bodies do different things and they become highly specialized. The sponges are the simplest and some of the oldest plants on the planet that accomplish that. So it's interesting. If we look at the package here, click and download that. I chose very carefully when I picked this cover, which I found online, it was very entertaining. Because if you looked back in time, and we talk about kicking it oldest school, well, these here are sea pens. Here are Medusa's jellyfish in their Medusa phase, and right over here we see sponges. They've been around for a very long time. Sand dollars, also known as echinoderms, down here. And I chose that cover for a very good reason. This is kicking at oldest school because we're talking Cambrian, Precambrian life forms here. Things on the bottom of the ocean that you wouldn't recognize as being animals if we really hadn't talked about it. So we'll get going with our sponges, and I've already got that page preloaded. It's just sort of here in the background. And they're known as phylum Porifera. Now, phylum Porifera is a very interesting term. You really want to focus in on the fact that it's porifera, like pores, and fera is a Latin word which means to bear. So these bear pores. Think about that for a second. Oh, sponges have pores or holes in them. They are the earliest multicellular animal that was and remains to be a spectacular filter feeder. They've sometimes been called the vacuums of the ocean because they pull whatever's around them through their body wall and scan it for food, they're filter feeding, and use it as best they can to help them survive and move on. And they've been very successful at it. And sponges are amazing creatures. Some of our greatest cancer remedies come from materials and compounds that we've extracted from sponges. Okay, so we'll get going with our sponge lecture. The course website has some things you absolutely cannot miss out on. Okay, we got a link to Tim and Moby here, our friends, and I'll tell you to click that online where they talk about sponges, and there's a quiz there for you. This may look a little repetitious, but it's showing a flash preview here. If you click on Anatomy of a Sponge, or look at its reproduction, it goes to a neat little animation, and you can sort of see there's different parts of it we'll want to go to. You'll see me go back to this throughout the video, and sponges, freshwater sponges, when they do a neat little sort of trick called producing gemmules to survive in harsh conditions, you want to go back to each of these, and I'll try to show as many as I can in the lesson. But I digress. We'll get started with sort of the notes content and give you something to fill in your notes package with. So again, when you look at the pictures that you see here, those pink critters right here are sponges. And that might look a little foreign to you because you think, hey, I know sponges. They're those things that, you use in the shower. They're really tube-like critters with holes in the side. So the sponges that you know are kind of like clumps. They're a slightly different version of this. But the water and everything that they want to consume that's in the water passes in them, and they have a current that they're able to create, and the water flows out the top, and they filter feed and grab whatever they can that's in the water column to help keep them alive. So here we go. Phylum Porifera, the pore bearers. Okay, so let's break down that term, periphery, and call it for what it really needs to be identified as. The pore bearers. There we go. And there's your terms, broken down. That's the nice thing about Latin. Helps to make learning biology a little simpler in that respect. So we're looking, of course, at kingdom Animalia. We go down to a major phylum, phylum Periphery, and these are some of the oldest animals on the planet. Sponges, in their adult life, you'll find just attached to the bottom, sort of glued to the bottom in a single spot. So the term that we use for that is we say that they are, if they're attached to a single spot, we say that they are sessile. And you just want to get to know that term. They're glued to the bottom right here. The reason why sponges fit into kingdom Animalia is for all the major reasons. Obviously, we're not looking at a plant or a fungus here. We're looking at heterotrophs. So they've got no cell walls like fungi or plants. They eat other things. They've got many cells, and they do start to become specialized. So this is nature's first multicellular heterotrophic specialized animal. Now, the animal part, which you're going to have a little bit of trouble seeing, will become more apparent once we zoom in on the cells that are inside of a sponge. And they're quite animal-like. Let's make that a little smaller. There we go. And we'll get to that now. Well, if you go hunting for body systems and you say, hey, where's the circulatory system and the respiratory system? And where's the reproductive system in a sponge? You look for like the organ systems in a human. You say, where are the equivalents in a sponge? Well, you're going to be sort of, well, let down a little bit. Sponges have a very simple system. And as we go through this lecture, you'll see why. Most of their body systems are accomplished, but they're just so simple, like a respiratory system. They don't really need one. Uh, they're able to take oxygen out of the water, and it diffuses into their tissues, and their waste products just diffuse out. The cell layers are so thin that actually works. And the interesting thing in nature is if you don't need a system like that, if you don't have to set up a system, why would you? So sponges, you say, well, most animals attack and kill and eat things. Sponges don't have a mouth or a digestive system. Actually, they do when they filter feed, bring things in. But sponges, you got to realize, are sort of Mother Nature's first multicellular experiment. They're cells that come together and they're specialized and they do these jobs, but we haven't built up to something as fancy as tissues or organ systems. It takes a little more time to develop, but sponges do this very well, even without these conventional tissues or organs. So, what have we got with a sponge? Well, first of all, when you think about a sponge's symmetry, they're very primitive. I'll switch out of purple. I'm getting tired of purple. If you look at human beings, we are bilaterally symmetric. Okay, so that would be bilateral. So that our left and right sides are more or less kind of mirror images of each other. Now look at another ancient creature, more ancient than us. We're going back in time now. And you look at our friend Patrick Star from SpongeBob. And let's change colors here just to make the point. This friend of ours has radial symmetry, like spokes of a wheel. So the body design is set up into pieces, which really traces a circular pattern, if you think about it, where each of these are radii. But every slice is basically similar. Okay, so like chunks of a pie. But when you look at a sponge is so oblong, and many of the sponges they don't even have radial symmetry. Now, a few more of the advanced members have a bit of radial symmetry, but a sponge, let's draw it from above, might look kind of lumpy like this. And looking at them, you say, well, which way am I going to cut that? Now, it's a crude drawing, but sponges are so lumpy that they're sort of not the same in any one spot. So you're like, well, that's not quite like that chunk. Those aren't the same. That's not quite like that. No, that doesn't work. You can't really say that this is bilaterally symmetrical on either side. No, that doesn't work. So you're kind of left with a case scenario where you say, okay, sponges really, because they're the most ancient animal, hadn't developed symmetry yet. The setting up of the tissues and more or less the body design was so primitive that the cells came together in groupings, but we didn't get anything that was symmetrical. Nice little undo feature, so I can sort of erase. Here we go. Asymmetrical, Latin word which means without symmetry. Because of this, you can't really say, well, okay, that's the head and that's the tail and that's the left side and that's the right side. Think about the basic dimensions. There's no front, back, and there's no left or right sides. It just doesn't exist. Not in the asymmetrical sponges. And remember something I've been mentioning in the course, the less symmetrical you are, like if you get back to more primitive symmetry, radial symmetry like you see with starfish, or asymmetry, a lot of those creatures tend to be able to regenerate. So they're asymmetrical. There you go. I've gone on and on about it. Body design is really quite unique. This points it out quite well. Our sponges are filter feeders. And what they're doing is we'll get into this in detail here they're pulling in water and anything in that water in through these pores. So I'll just sort of highlight the edges of these in blue so you can see them. So they're pulling in water and anything extra it has, so little food particles, you could say there might be algae, for example, and if that gets pulled in, they can grab it and use it as a food source and they can consume it. So they create an interesting current by pulling in the water, and that current, let's trace this a little bit, passes out the top, and this top, they've done that sort of a cutaway view for you here, but this top is known as an osculum, and that's a term you would need to know, but we'll get into this in detail in our pictures. And there's a great number of cells here you're going to want to be aware of. In fact, you can see them in the background, and we'll get into them. These little cells that you see right here, there's some interesting anatomy in here, this little spiny shaped thing, and a little sort of amoeba shaped cell in there. So these cells in the middle, closer to what we would call the spongocoel, also known as a lumen, which basically means an empty cavity. These cells along the side create a beating pattern upwards with their flagella, which I'll just draw like this, and that creates a current which draws the water upwards. So we create this net directional flow like this. I love that zoom in feature. Look at how small I was writing once you zoom out. Okay, so the large central cavity, and what they're referring to here, central cavity. Any hollow space is usually known as a lumen. This has been called many things: central cavity, lumen, In fact, when you get into Biology 12, they'll talk about the hollow space inside of your blood vessels as being the lumen. So there's some synonyms here that you'd want to know. Okay, but it's just the space that the water is going to pass through and then pass out of. Now we're looking at several sponges here. We have sort of a larger one in the center and a couple little ones on the side. But this is why they're called the vacuums of the ocean. Let's make that A a little better. They're called the vacuums of the ocean because they're continually pulling in material. So they're cleaning the water column. So that's an important ecological role. They help to keep the oceans clean. Okay, so now we start to add a little bit of extra information, a little further detail. And I've sort of alluded to or referred to this a little bit. Here's your osculum. There you go. And the water flow, of course, is well defined here. Sort of like a one-way water flow. And the pores are found all around the outside of the sponge. They've just drawn them a little bit larger here so you could see it. So I'll outline those again with a little bit of coloring. So, enters through the pores and leaves through the osculum, which is a big hole at the top. Now, zooming in on what actually makes the water current, these cells, called choanocytes, which I'll highlight here, have a synonym. There's another name for them. When I learned about them, they were referred to as collar cells. Reason why? They have these little collars. So let's change up our color here a little bit. Maybe a darker color for a bit. They're called collar cells because as the cells face into the central cavity or the lumen, there we go. There's your collar cell right there. There's its nucleus. There's the flagellum of that cell. And that flagellum, as I referred to, is beating. They're all beating in more or less an upwards direction. And that's creating a water current. Like that. Now these cells, there's the little collar basically from there to there. I'll just undo that so that doesn't stay. If you say choanocyte or collar cell, I'm okay with it. Either way. Collar cell's a little older, so no wonder I want to use that term, right? So these cells are quite specialized, and that's what you're seeing. Like, sure, we've got cells in the sponge that do different jobs, but now you start to see that there's cells that are different, and that is the whole essence of being in kingdom Animalia. Multicellular, specialized cells, eating other things, all kind of goes together. Now, sponges, there's several classes of sponges that you'll study, and I'll outline these in a minute. Sponges are classified by the little hard parts in their body wall. You really don't want to call them bones. They're known as spicules, but they give them structure. It's a little spike. Now, it can either be made of calcium carbonate, which is CaCO3, so let's write that down. Calcium carbonate. Or something, it's a hard term, most kids won't like this, but silicious, like silicon dioxide, like glass. Calcium carbonate, glass or glass-like, is basically the content of our bones. It's an interesting little spike. It's right here, and you'll find it. Now, not all the spikes are shaped like shurikens, throwing stars. In fact, you can see all different types of kind of shapes, and that's more or less what this picture is here that you're seeing, that I'm outlining right now. There's a whole variety of shapes that we find depending on the sponges that we're looking at. And it's a really useful way of classifying them. And I'll get to that, but there's four major classes based upon the spicule variety that they have in the wall. But picture this: there are glass sponges out there where they've got so many of these spicules in their basically in their wall, right, and in the exterior. It's not a cell wall; they're not plants, but basically in their sort of hard outer skin, if you will, that they actually look like they're made of glass, but they're filter feeding, and they're animals. It's pretty strange when you see an animal that looks like it's got a glass body, but that's exactly what we're talking about here. Now, what makes all this stuff? There's sort of a primitive cell inside of the sponge, which can become anything, and it's called the archaeocyte, and it's an amoeboid-like cell. Archaeocytes. So let's see if I can get a new highlighter color, because I think it's time for a new highlighter color. Let's go with like a nice with a sort of a pinkish color. So our archaeocytes move around in the wall of the sponge. Let's see if I can zoom in. There's not a great picture of them here. Let's see if they... Nope, they don't come up there. I'm going to zoom back. They are there, but they're just a little bit hard to see. Here we go. This cell right here, you might have noticed sort of hanging out, is a little bit ill-defined. It's pretty simple. It's really basic and primitive. There's another one down here. There's its nucleus. These cells are capable of giving rise to just about anything. They can create other archaeocytes, they can make the choanocytes, they can make these spicules. These cells do it all. So there's our archaeocyte right there. So we'll just put arch for short. And they exist kind of in this middle layer. And it's their job to give rise to new cells, to make spicules, and to literally keep the cells all fed. They do so many things. They feed the cells, they give rise to new cells, become cells. They clean up the mess in between. They've got a big job to do. They can become anybody, but they're sort of responsible for so many activities: cleaning and feeding and becoming things. And they're sort of a jack of all trades, if you've ever heard that term before. They're capable of moving around in that inside layer and doing all these jobs that they need to do. Now, body systems. Let's break it down. What do they have for body systems, period? So feeding, and I'll superimpose my will on this and say, okay, feeding, that makes me think digestive system. Their mode of feeding is that they are filter feeders. Pretty straightforward. They're heterotrophs, but they're filter feeding to get other things to eat. It's the choanocytes, as water comes in through the pores and the choanocytes create the current. The choanocytes, more or less, you can think of the food particles as kind of getting lodged on them, and the amoeboid cells will get a hold of that food and move those nutrients around where they need to go, keep everybody fed. So food particles trapped and engulfed by the choanocytes, and then the amoebocytes move in. The choanocytes would make sense to be the ones to pick it up. They are the ones that are doing the filtering. If you look back on the choanocytes, you'll see that they have a neat little collar right there. And it's that collar right here that helps to pick up and trap all that material. There's your cell nucleus. Get the idea? And there's a little bit of a pass off of materials to our friends in the middle. Reproduction. Interestingly enough, it's kind of shocking. It's internal fertilization. Now, there's animals out there that are more primitive than us. Humans practice internal fertilization, so it would seem, if you think with mammals, that's kind of advanced. That's happening a little later in evolution. Sponges did this forever ago. Even fish don't do internal fertilization. Look at fish. They the female fish lays her eggs and the male fertilizes them. Well, that's external. That's much cruder than what humans do, right? And a lot of mammals. Sponges were doing internal reproduction millions and millions and millions of years ago. And it's quite simple. Sponges can decide to reproduce sexually or asexually. And the picture that you're seeing here is the release of sperm. So this is a sexual phase. That's a male sponge. So let's make the note. They've got the asexual or sexual option, which should not really come as a surprise. Since sponges are so simple, they could just choose to bud. But if they want to shuffle the genetic deck, they can do sexual reproduction too, which is good for keeping the genes mixed in the population, right? There is fertilization inside the sponge's body. You can think of that as female, which gives us the whole concept of internal fertilization, which we'll look at now. Actually, let's pop back. Okay, here's where I'll go to some of my animations because they're really useful at this point. And in fact, if I was wanting to prepare for a test, I'd run through the animations. A couple of these ones are great. They're just freely available on the web, but I want to show you what they're more or less all about. Again, these thumbnail previews are functional, but they're really annoying. They're too small. So let's go to a flash video. There we go. And more or less just kind of meet the sponge. So since we're talking about reproduction, I'm going to talk about reproduction, just kind of work my way back a little bit. So here's sexual reproduction, since we're sort of talking about that. In sexual reproduction, the sperm will be released from the male sponges, and they're produced by cells inside of the sponge and released outwards, and the current that the sponge creates carries them up, and they're just looking for another sponge nearby to fertilize. Now, if that sponge pulls in the egg or sperm, so we'll just pause that, there we go, they go in through, let's see here if I can zoom in a little bit. No, it's not going to let me zoom. They go in through the pores on the side. Passing through, now we're kind of zooming in, they're capable of fertilizing. Now this is interesting. You've got basically the equivalent of an egg in the body wall of a sponge. So it's just waiting to be fertilized. And there we go. You get another diploid organism and it's dividing. And that's birth right there. It just got carried away. So the larva gets carried away. And when it lands, it will grow up and become a genetically diverse sponge from the parents. Hit reset. Now, if sponges are just looking at doing asexual reproduction, then a bud can just form off the side of a sponge, like this, and just drop off. Very simple process. Budding, genetically identical to the parent, and that grows up. Now that'll happen if there's very little stress in the environment, and it's a good time to just create clones of yourself. Let's back up for just a moment to digestion and look at obtaining food. And this is a great, just a tremendous opportunity to examine this. So what you've got here is you've got your collar cells, also known as choanocytes, and you can see their beating motion. There's your spicule, which identifies different classes of sponges. And there's your archaeocyte, also known as your amoeboid cell. And of course the pore going through towards the inner cavity. So there's your filter feeding. There you go. And you can see these particles are all coming in here, and they're getting more or less trapped by the collar cells, who are just kind of like little fishermen trying to pick up particles. And you see them, you kind of saw them slurp those particles in. Now waste cells, the collar cells, of course the nutrients they're going to get picked up are going to be passed around, and the amoeboid cells are going to help move those around. Waste from the sponge will just be released more or less by diffusion, because this water is a lot less concentrated in waste than it is over here. So they sort of release their waste products. It's not really poop or pee, it's just junk, carbon dioxide waste, ammonia waste, and it passes right back out the lumen. And if you look over here, that waste is just passing out the top of the sponge. So that's pretty typical sort of animal behavior. This is a great animation. Go back to it, get to know your sponge anatomy. Okay, so zoom. There we go. Kind of get back to sponges there. I kind of lost it. Couldn't resist with SpongeBob. Interesting how this chapter is sponges and cnidarians. There's our cnidarian up there. There's a third sort of reproductive strategy you see in some of the freshwater sponges. I should show it to you right now. Some freshwater sponges will go into a survival mode. Imagine it's basically doomsday for the sponge. In fresh water, the water could freeze, and the sponges, what are they going to do? It's getting, how do I survive? I got to make it to next year. Some sponges will produce something called gemmules, and this really nails it. I'll let the narrator kind of take over here, and then I'll just chime in a bit.
When living conditions become harsh, such as in times of drought or cold weather, some freshwater sponges produce internal buds called gemmules.
Okay, so it's asexual reproduction, but it's survival mode.
Gemmules are clusters of amoebocytes encased in protective coats. Spicules on the coats help protect the gemmules.
So they set the spicules around the outside, they set themselves up with some food in the middle, and these little amoebocyte cells in the middle are basically our archaeocytes, because they're what we call pluripotent. They're capable of producing cells of pretty well any variety that's needed to recreate the sponge, which is why sponges are so capable of regeneration. You can take a sponge and blend it up in a blender and think you've killed it, and if these cells survived and you give them basically a place to live and some nutrients, they'll regrow the sponge on you. So this is a gemmule capsule that you see with freshwater sponges.
Sealed in with ample food, the cells inside the gemmules can survive even if the parent sponges die. When conditions improve, the sponge cells emerge from the gemmules and grow into new sponges.
So there you go. There's a nice overwintering process, a survival. So great little video, and one of the things we'll do in class is let you look at slides of sponges in their different stages. One of the more interesting slides is if you get your hands on a gemmule, and if you slice it really thin in cross-section, you can see all the little amoebocyte cells in the middle, and you can see how it's more or less set up as we just described. Okay. Okay. There's some neat stuff here I'll come to at the end. I just wanted to make sure there wasn't anything I missed. All right. So back to our, just finishing off our slideshow. I'll just go back into reflection here. And we'll make sure we have all the content. There we go. So sexual reproduction was kind of where I left off. And I said that they can bud asexually. Freshwater sponges might produce gemmules. Sexually, we can look at internal fertilization. Moving on. Sponges are indicators of health. If you see that sponges in an area can't survive, you have to ask yourself a question: what is in the water that's killing them? When you look at some of the things that get dumped in the ocean, think about oil and poisons like cyanide, and just what's ending up in there. So they're indicators of marine health. They're also ecosystems for little things like snails and sea stars and shrimp. If you're going to be running away from a predator, that might be a good place to go hide out. Maybe they can't get you in there, right? Let's see here. Yeah, great place to hide out. Maybe you could fit into a large pore of a sponge, get away from your attacker. So that more or less takes us to the end of chapter 26.2. What comes up next is 26.3 in the cnidarians. I suggest you read ahead because there's some major learning to go on with those. But I should, before I leave, label what our major classes of sponges were. So I'm going to use a little bit of space here and zoom in like this. And what happens with your textbook, they're treating it in a very general way. But sponges, which is phylum Porifera, are broken down into classes. So you've got class Demosponginae, which is a traditional sponge that is more or less rubbery. The spicules in their walls aren't hard at all. Class Hexactinellida, that's a big one, which are known as our glass sponges, and they have silicon dioxide, little spicules. Again, that's the little shuriken thing that you guys had seen in the basically in the wall of the sponge. And there was a third class, third principal one that I want you to know, which is class Scleral Sponge. And not to be outdone, sorry, I forgot the fourth class, Calcarea. Sclerospongiinae has a variety of siliceous particles in it. So we'll say silicon variety. So push that out of the way. There we go. And Calcarea is really our calcium carbonate variety of sponges. So really, before I go, I want you to look at these ones so that you know, kind of what these major sponge classes look like. So a trip out to basically the web, take you there pretty quick. Oh, apparently can't spell. There we go. Let's go to images. Okay, so Demosponginae, I really want to show this. This is the kind of sponge that you would find, for example, in a typical bathing house, if you want to call it that, bathrooms. You still see them. There's scrubbies hanging off handles. People are working themselves over with these things, right? Just your typical variety of sponge. And when you go through different examples, you start to look a little bit weirder, but they're basically the rubbery sponge variety. So that's Demosponginae. If you look at Hexactinellida, there we go, comes up pretty quick. Images. These are the glass sponges. They can be quite amazing. Now, don't expect them to just look exactly like glass. It's just that they have a glass-like exterior. So that right there, glass tends to form a crystal. So they're very pretty, and they're still doing the same trick of pulling water in through their walls and filter feeding. But they have very sort of crystallized like bodies. That can be very nice. And their crystals set up in a very nice pattern, which is more or less what this is trying to show. Sclerosponginae, there we go. Sclerosponginae, let's go to the images. Now, because they have a variety of siliceous particles, you can get some ones that look really glass-like. Okay, Hexactinellida is glass-like, but Sclerosponginae just has so many glass-like particles. I mean, look at that. That's an animal. And they're getting away from Sclerosponginae, but I'd be happy with showing that off as an example. just very attractive version. Um, cal, um, Calcarea. Now I have to put sponge in. There we go. Calcarea sponge would be a very hard variety of sponge, like this. Let's zoom in. Pretty neat. Looking at this, you're like, well, what is that? Well, it's basically a filter feeder. And I wish I could zoom in a little further there for you, but you can see the little holes in the body wall, and you can see the holes on the top, which are oscula, where the water would be flowing out of. So pretty cool looking sponges. And again, these ones have some pretty interesting spicules because calcium carbonate forms a crystal a little bit different than silicon dioxide does. So you get different body appearance. More or less, that's what makes or controls the body design. So there's our four major classes. Better late than never. But now you really know what a sponge is. Nature's first multicellular animal that's a filter that was and remains to be a filter feeder. Their impact is quite significant. In fact, I won't run it in real time, but what I will tell you is that the sponge industry, sponges themselves are a tremendous economic boon. And in fact, in the Aegean Sea in Greece, people would go out and collect sponges underwater, and they would sell them, and it was a very important source of economic viability for people. This is how they made their money. And off the south coast of Florida, it's also extremely popular. The sponge industry, there was a very large Greek community that had transported themselves to southern Florida, and what they had done is continued what's called sponging. Now those videos are here. So deep sea diving, actually I'll update this, this should say in Florida. And British Columbia, as it turns out, take a look at that, has one of the world's rarest glass sponge reefs off the east coast, more or less, of the Haida Gwaii, also previously known as the Queen Charlotte Islands. So a neat little news report from CTV. It's a little link out to YouTube there for you to take a look at. But I'm sort of hitting my limit here. Quite a bit of information on sponges. Good thing about me being recorded, if you get bored, you can fast forward, rewind, or just stop me altogether. So folks, that's it. I'm down to 63% battery, so I better check out. In the meantime and in between time, take her easy out there. Ciao.